Experiment 3 is all about molecular absorption spectroscopy. So you identify a substance by the color or wavelength of the light that is transmitted. So this is just an illustration of how this works. So if we have a white light coming into a sample, and white light consists of red, orange, yellow, green, blue, and violet. If we have white light coming in, and for sample 1, if we have white light coming in and what we see is black, that means all light is absorbed in this sample. Sample 2, if white light is put into the sample and we get out white light, then we see that no colors are being absorbed in the sample. Sample 3 is a little bit more complicated. So say we have white light coming in and what we see is violet. So something is being absorbed. So we then look at the color wheel in order to identify what's being absorbed. So the color wheel, the way we can tell what's being absorbed, what's not being absorbed, is that a solution is the complementary color of the light absorbed. So whatever light is being absorbed, the complementary color is the solution. So in the example we saw white lights coming in when we see violet. So if we look at violet on the color wheel, violet is complementary to yellow. So in this example, if you see violet, then the color absorbed is yellow. If we see red, then the color absorbed will be green. And vice versa, if we see blue, the color absorbed is orange. And these go reversible. So if we see green, the color absorbed is red. If we see yellow, the color absorbed is violet. And that's how we determine our solution. So in this lab, we're also going to cover transmittance, which is a relationship between the intensity of the light coming out of the sample and the intensity of the incident light. So here's just an illustration. If we have a light source coming in, this creates the incident light. This is a sample, and then the transmitted light is the intensity. So transmittance is the intensity of the light coming out over the intensity of the incident light. So if no light is transmitted, meaning we see a black light, we see black coming out, that means that T is equal to 0. So another relationship between incident and transmitted light is called absorbance. So absorbance is given by the equation negative log of T. So if T is equal to 0, then all light is absorbed and A is infinity. If no light is absorbed, then A is simply 0. So A equals Kc is an equation relating concentration to absorbance. So K in this instance is a constant dependent on the wavelength, and C is the concentration. So as concentration goes up, absorbance also goes up. So more light is absorbed when the concentration is increased. So I wanted to quickly go over a dilution example problem. For this one, it asks us to calculate the concentration of 500 milliliter sulfuric acid solution prepared from 10 milliliters of 17.8 molarity of sulfuric acid. So we're going to start by using this equation here to relate and to find the concentration here of the sulfuric acid. So if we start, we know that we're solving for this, the concentration, and we have 50 milliliters of sulfuric acid, and we're relating it to they're already prepared 10 milliliters. So we have 17.8 molarity of sulfuric acid with 10 milliliters of the solution. So in order to solve for molarity, we just divide this over. So we get molarity is equal to 17.8 times 10 milliliters divided by 50 milliliters. So our answer ends up being 0.356 molarity of sulfuric acid.